All right. Hi, everyone. My name is Malaz, and today we will be looking at some biochemistry passages that we'll do together and just go through them and try to answer some questions. Just a little bit of information about us. We are a nonprofit organization, and basically we're all volunteers, and our goal is to help students achieve their goals in the MCAT. So if you have any questions, feel free to email us at info@premiumpeers.org. If you're also a student in our Slack channel, feel free to reach out to us via that source. All right. So a couple of things I thought I would start with to mention, especially focusing about what strategies we're going to use today. So the strategies that we're going to use today while reading the passage, and feel free to use your own strategies, just a way that we can be on the same page when we're doing this. So we just really want to get the general idea of each sentence and each paragraph. We don't want to get bogged down by all the details they give us. We just want to get the general idea of what's happening. What is really important, though, is when we encounter experiments or figures, we really want to break them down. And so what we want to do is we want to break them down by these three categories. What independent variables? What are the dependent variables? And what is our pattern? So the independent variable is the variable that we are changing. This is usually our x-axis. It could be different. Dependent variables are the one usually on the y-axis, and they are what we are measuring in our data. And the third thing that we look at is pattern. What does our data tell us? The strategies that we're going to use to answer our questions is this. So before going to an answer choice, we want to find a guide. So what I mean by guide is sometimes either information that you already have, or could be a sentence from the passage that we would like to use to help us find the right answer. So before we look at the answer choices, we get an estimation or like a guided estimation of what his answer is going to be, and then we go with that and we pick the answer choice that best goes with our prediction. So the reason why I'm mentioning this is always the right answer is in the passage. So if we go with a prediction from the passage, the right answer is going to be the one that corresponds to that. The second thing we're going to do while going through answer choices is we're not going to read four at once. We're going to read two and compare them to each other, and then we're going to go down further. So like using a process of elimination. All right, just some general review before we do this passage. Basically, this is an amino acid chart brought from Jack Weston. And what is really important for the MCAT, what they focus on is the group of each amino acid is in. And so we have general categories for amino acids. So I'm going to go through them through this diagram. So we have non-polar amino acids. These could be aliphatic, meaning they're not cyclical, like aromatics shown here. And so these amino acid correspond to this non-polar aliphatic R groups. Other ones could be non-polar and aromatic, where you can see them over here. The polar ones that are uncharged or have a neutral charge are located over here. And then we have the positively charged one and negatively charged one. Identifying the category of each amino acid, especially knowing the one-letter code for each, is really, really important. So we would want to see the one-letter code and be like, oh, I see a D. What is that? A spot. It's negative. And that should be within 2 or 3 seconds. This is really important. Okay. The second thing that I want to talk about today is the structure of proteins. So what are the structure of proteins? So we have a primary structure. Basically, this is the amino acids, individual amino acids combined together to resemble the total protein that we have. Then we have a secondary structure, basically is the local folding of the polypeptides using the skeleton of the amino acid. Then we have a tertiary protein structure. So what is this? This is really important. This is due to the side chain interaction, not due to the skeleton interaction. And so what is happening in this, think about it as the protein folding, right? And the other one that we have is a quaternary structure. So when you hear the word quaternary, we think about multiple subunits coming together. This is when you hear the word ternary or a monomer, a dimer protein. This is referring to the quaternary structure. So the second important thing that I wanted to bring out is really important to know which bonds stabilize each structure. So the peptide bond stabilizes the primary structure. The hydrogen bonds stabilize the secondary structure. The tertiary and quaternary structures are stabilized by very similar bonds. So there are non-covalent bonds that stabilize them. These are hydrogen ionic or hydrophobic interactions. And very importantly is we have this covalent interaction called a disulfide bond that do stabilize this. So I just want to add one last thing really shortly to summarize. The tertiary and the quaternary structure is stabilized by side chain interactions. These two are stabilized by the skeleton interactions, which are different. All right, moving further to this figure. All right. Um, all right, so this is the tertiary structure that I talked about, and it's just resembling the type of bonds we just mentioned. We have ionic interactions, we have hydrophobic interactions, we have disulfide linkages, which is covalent, and we have the hydrogen bonding. So if you get a question that is asking you disturbing what kind of interactions would disturb the tertiary structure or denatures the protein, the answer is going to be something about disturbing hydrogen bonding, disulfide bridge formation, or ionic interactions. All right.
Can I ask a question, or should I?
Of course,
Wait.
go ahead.
Oh, I was gonna ask because I've seen questions where it's you raise the temperature and like so one of the questions I've seen is when you raise the temperature, that does the enzyme activity or its performance increase or decrease? And I know the general rule of thumb is that it increases, but at some point it goes down because it denatures. So if we switch back to the question of temperature, like which one, if temperature were to increase, which one of these would get affected first?
They would usually tell you, they would tell you the temperature is very high or if it's not very high. But generally, to go with is increasing temperature increases the enzyme function, right? This is enzyme kinetics. But
Right. Yeah.
generally, when we're talking about protein structure, when they mention the term temperature there, increasing the temperature, it's usually in the context of denaturing the protein.
Okay, got it.
So it depends on the context. So if the temperature usually mentioned in the protein structure, it's usually denatures it. But if it's mentioned in a context of enzyme kinetics or reactions, it usually increases it. But they usually will give you more information about that because if not, you can sue them.
Okay, thank
Yeah.
you.
All right. Okay, so I'm gonna start with this passage. So I am going to share this tab instead. Um, so would you like to try reading the first paragraph? You can read it out loud or in your head, and you can tell me what you got out of it, or do you want me to read it? What do you feel comfortable with?
Um, I can skim it and just tell you what the takeaways are, like
Okay.
in my head.
Yeah.
Let's see. Okay, so I'm probably missing a couple of things, so you should do that when I read this. First thing, autosomal dominant. So if they have any questions about like
Mm-hmm.
the
Mm-hmm.
Punnett squares and whatnot, then something to consider.
Mm-hmm.
Let's see, polyglutamine. So glutamine, I believe, is polar.
Right.
And then, but not charged or anything. And then near end terminal exon, HTT. I mean, I got exon as post translation, post transcriptional. It's not going to be like an RNA transcript would reflect it. Plus one toxic conformation. And then the last part, I think I'm not interpreting that right.
That's okay.
End terminal, what would you, the end term, there's one thing about configuration, it is something I'm taking it out. Like,
So, so you mentioned a really important piece is they tell you what is it. It's autosomal dominant. The second thing that is really important is, okay, so we have polyglutamine. Basically, we have a lot of glutamine, right? So where in this protein, okay, this information, I really don't need to focus too much on it. This is what I mean by technical information. Near the N-terminal exon 1. Like, this is the location of it. This is the size of the protein. It's just additional information. But the big picture is, okay, the Huntington protein, right? We can have a disease if you have a lot of glutamine on it, okay? And this next sentence tells you, what does this do? It affects the protein-protein interaction. So having a lot of glutamine on this protein will affect the protein-protein interactions so that we will have this disease. Does that make sense?
yeah.
Do you see how I'm going big picture on this?
Yeah.
Okay. If the questions might ask you about the details, this is 100% valid. But what we really want here is we do want to just to get the general idea of it, because we don't have time to really understand every detail. All right. Do you have any questions before we move on?
No, that sounds good.
Okay. Let's take turns on this. You read a paragraph. I read a paragraph. What do you think?
Like out loud or?
No, if you what you did was fine. So I can try reading this paragraph, and we can see what we understand from it. So the folding misfolding of this protein fragments provides insight into the production of pathogenic amyloid-like aggregates. So
Thinking
something about
Alzheimer's.
something about aggregation that is affecting it. Remember in the last paragraph they talked about pathogenic interactions. Now they
Oh,
clarifies it. It's aggregation.
okay.
Okay. So figure 1 describes a model which summarizes current results on amyloid fibril formation. I don't have to look at it yet. I just keep reading. Protein folding step 1, okay, this one, begins proceeding normally, okay. However, it appears that the polyQ amyloid fibril initiation, step 3, takes place only after the formation of oligomers liking beta structure, step 2. I am not understanding much from this, but it seems that step 2 is important. Once fibril formation has been initiated, the polyQ regions forms highly rigid beta structures. Okay, so we first form the fibril formation, and then we form the rigid structures. Does that make sense?
Yeah.
Okay, which is shown here. And so you see we add them on each other, and then we form the beta barrels, and this is what we have. Okay. Do you want to try this one?
Read it out loud, read it.
It's up to you, whatever you feel comfortable.
Okay. So I guess I'll go through sentence by sentence. SEC is I mean, it's based on size, and then the larger ones get out first. So keep takeaway. HD separates. Okay, so monomers, tetramers, higher order oligomers. So monomers are going to elute last, presumably. Dependence of HTT exon 1, like fragment size on length. Okay, so dependence means it's X is probably going to be our fragment size, and then Y going to be the repeat length, I think. We will see. And produce fragments containing the exon, and then assess their size. Okay.
May I
No,
interrupt a little?
yeah. I'm getting
Okay.
into technicals. I'm aware.
Yes, let me help you a little bit. So what's really important from this paragraph is actually this sentence, these two sentences. So they are describing what is it, right? So the wild type is monomeric. But look at the next sentence. Fragments isolated from HTT protein isolated. This is a terrible sentence. From HD sufferers. Basically, the mutant protein tend to form monomers, tetramers, and higher order oligomers. Do you see this now?
Oh,
So
okay.
there's a difference. The mutants not only form monomers, but also form tetramers and higher order oligomers. Does that make sense?
Yeah.
Okay.
So that's going to differentiate. Okay.
Yeah. So we move on. So therefore, researchers assess the dependence. So this is long sentence. I would just call it the protein. So looking at the protein size on glutamine repeat length. So they're saying what does the effect of increasing glutamine on the protein. So they produce two fragments containing the N-terminal, okay, the protein, and 23 and 37 polyQ repeats, and assessed their size. Number of monomeric subunits when fragments were added to a buffer. Okay, this is additional details. So what's happening is made two proteins. One has 23 glutamine, one has 37 glutamine, right? And they're looking at the number of monomeric subunits.
Okay.
Okay. All right. Let me ask you this question. What do you think the mutant protein will have, more or less monomeric subunits?
It's going to have fewer or less.
Yes,
Wild type's going to have more monomers. So in size
exactly.
exclusion, well, actually, I shouldn't jump to that, but in the context of size exclusion, wild type is going to elute last. Mutant's going to elute first.
Let's keep it big picture,
Okay.
right? We don't want to
Okay.
go to that into that. This is how we can save up on time. And
Yeah.
so what's happening here basically is, okay, we have two proteins, one wild type, one mutant. The mutant has less monomers, right? And this is what they're trying to see. How does increasing the number of glutamine, because the mutant has more glutamine on the size of the protein? So this is our expectation, right? So let's see the next sentence. The fragments were monitored by specific type spectroscopy, which measured fluorescence dye covalently attached to the sulfohydryl side group amino acid. Technical details, we might go back to them, but they use some form of a test, okay?
Okay.
Let's see. So they give us this figure. Okay, let's see what's happening. So, and look at the x-axis. Okay, they're increasing the concentration. Okay, what is the y-axis? They're measuring the size of the protein or the number of monomeric subunits. Okay. Now, could you go ahead and tell me what do you see next?
I think these log curves? Are these log transformed? I mean, I don't think it's at this point. So, okay, it looks like they're increasing the concentration of the glutamine repeats. And then based on that, you're going to get a number of like monomeric subunits. I don't even know if I'm interpreting this right. So let's see. As the number of 23 glutamine repeats increases, size you have a a size jump, but not considerably compared to 37. I'm not sure if I'm interpreting this right.
Let's keep it even more big picture. Okay.
Okay.
23 is the gray, right?
Yeah.
37 is the black, right?
Yeah.
Which one has which one is smaller in size, the gray or the black?
Be the gray, right? Because fewer
Mhm.
monomers.
Yeah.
Okay.
So the gray seems to be small. Does the black have any monomers?
It starts off. It has four towards the end. It has like 4 or 5 monomeric units, right?
It starts on
Subunits.
2. Is
Yeah.
if we have 2 monomers, what do we call those?
Oh, dimers.
So does it have any monomers?
Oh, you're. Oh
Do you,
no, then.
do you
Or,
see what I
maybe it
mean?
has. Oh, is it? So it just has a dimer, not a monomer, or
Because 2 monomers is dimer, right?
okay.
So it tells you the number of subunits, okay?
Yeah.
So if you have 2 monomeric subunits, you have 2 of them binding together. You have a dimer. This is what it is. In this one, you can see this. So it varies on concentration. But in general, the general pattern is we see the smaller size is for the less glutamine, right?
Right.
Okay. Would you like to try question 1 for me?
Okay, I'll try talking it out. Let's see. Toxicity is greatest when it contains. So we're looking. If it says toxicity, we're aiming for the mutant.
Yes.
So we preferably not want monomers. Or sorry, yeah. So lack of monomer. So it's my hunch is it's either A or B.
Okay.
Just yeah. And
So
then this greatest one, and then 37 at the lowest concentration. I already had a dimer, so I'm gonna guess it's that.
do you think the answer is A?
I think so, but I don't feel confident because let's see. Yeah, we can give it a go.
Oh, what? You want me to sign in? All right. Let me sign in really quick. Hopefully it can work. Let me open it again. Okay, so I'm here. Okay, you got it right. But why were you not confident in it?
Because I lack confidence through this whole thing.
Oh no. Like I mean, like sometimes like
j-
some answer choices can be definitely wrong, right? Like
so I could just walk through so I think my interpretation was initially wrong, and then you kind of guided me in the right direction. So that was, I think the first thing was I knew it had to be a lack of monomers because that's kind of what makes the mutant deadly to begin with. And then it was just a matter of comparing the curves. So
yeah, exactly. So yeah, that's it. All right.
okay, so which amino acid mimics Huntington's? The only thing really mentioned here is glutamine. So is it literally is that one, two, totally one? No,
Make
okay,
it
so hold
smaller.
on. One, okay. A is asparagine. One, two, three. Okay, B is lysine. That's tryptophan. Wait, did I get this? Go back up. Sorry, I don't think I read the question properly.
Oh, I'm sorry.
Repeat it. Wait, is mimic? Oh, mimics. Okay, so my guess is. Well, let me. Can I go to D one more time? I think it's A, but okay. I think it's A.
Here you go.
Yeah. Okay.
Nice. So doing great. All right, let's do number 3.
Based on the model. Oh God. Okay. Good. Can you scroll to the? I have to go to the model again. Primary structure can only be, well, even without So even without just even looking at this, primary B doesn't make sense in. I'll still look at it, but B doesn't make sense in. B and C don't even make sense in general. Let's actually let me go back. Okay. Based on the figure one, cylinders and arrows represent B. Okay, let me just talk this out. Sorry. So protein folding, step 3 takes place only after the formation lacks beta structure. So poly-Q initiation, they lack beta structure, so they don't have a secondary. So what exactly is step 3?
Okay.
So this is, it involves, so basically based on beta, my guess is immediate. Okay. Sorry.
Okay. Are fibrils tertiary or quaternary?
Oh, it's sorry, can you go back up in the passage? Expansion. Those poly-Q is beta structures, so I think it's secondary.
Okay.
They call poly-Q. Why am I not getting this at all? The poly-Q region is beta structures, so it's second. I think it's secondary.
Okay. Let's go to the answer choices just to make it easier, right? So they're talking about something forming before the other, right? In all of them. Is that right?
Right, and I think based on that we can eliminate B because beta. If they talk about beta structures, we're looking mainly at secondary, and part of the problem here is it lacks.
So we can remove B and C, right?
Appears initiation. So
okay, now, okay, let me make it easier for you. What do we see in the beginning?
Cylinders, alpha, arrows. So those are, if those are alpha helices, that would be secondary.
But what is this?
Oh, that's a polymer. So that would be a.
Okay, let's
That would
make
be,
it
sorry.
let's make it even easier, right? Let's go to this sentence. What does it say?
The poly-Q version. So step 4 is oh, so that's, oh, so it goes to secondaries at the end. So
Correct.
it's completed after something else. So that would, I guess it has to be A.
So that something else is
No, complete.
febrile,
Wait,
right?
sorry. Wait, sorry, go ahead. Febrile, and then that, if you have a bunch of repeating monomeric units, I thought it would be quaternary.
Is
But
that
I
tertiary or quaternary?
thought it was quaternary, but I'm starting to
Correct.
doubt myself. Okay.
Tertiary is folding. Quaternary is combination of multiple subunits.
Okay, so
Since febrile is the combination of different subunits, it's a quaternary. So we can answer this question in an easy way is we can just look at this sentence, right? Once febrile formation has been initiated, so febrile comes first, then we form the beta structures. So the beta structure doesn't come till the end, right?
yeah.
So this is it. What we formed before it is here. You see N here. When you see this number, usually means you have multiple subunits combined together. And from this, our assumption could be then it's A.
Okay.
Does that make sense?
Yeah.
Do you see why it's really important just to go to one sentence and just stick to it so that we don't confuse ourselves with too many things happening?
Yeah, that's part of my problem.
It's a skill. We can all work on it. We
Yeah.
gotta try to stick to one sentence or find where is our best clue and just stick to that. All right.
Based on the ribbon diagram in figure 1, the beta sheets are, oh, I can't even, okay. So the arrows represent, well, I'm going to try to make the screen a little. Beta sheets of poly HQ are, when it says arrows represent beta structure, can you explain that
Yeah.
a little?
What do you mean?
It says arrows represent beta structure.
Mm-hmm.
What is that exactly? Like which arrows?
These arrows.
Which one? Oh, okay. I thought, okay. So can you zoom in a little? Sorry.
Of course. Oh yeah, it's really hard to see like this. Here you go.
Okay. Okay, so they're anti, I think it looks like they're going in opposite directions, so I guess it's anti-parallel.
Okay.
And usually in that, it's just because it's the primary structure, it's like different end termini, like they're in different ends.
Mm-hmm.
So I'm guessing D, but it's also when I'm kind of on the, I don't know if I'm interpreting this right.
Okay. So you're stuck between either C or D, right?
Right.
But the difference between C and D is one it says they're aligned, one it says not aligned. Okay. If they're anti-parallel, do you think it's going to be aligned or not aligned?
Not aligned.
Okay, let's check. And you're right. Does that make sense?
Yeah.
Okay. All right.
Last, can we go to the end? That's the paragraph that mentions it. Sulfhydryl, so it has to have some sulfur. So cysteine or methionine, right? Then a so it's most likely cysteine, especially with all the beta mentioned about beta structure, if I have to
So you
guess.
think it's C?
Yeah, I think.
Yay. All right.
The rigidity. So B is not true because no ion, nothing's charged.
Okay. Let me ask you a question. What is our guide? What is our prediction for this answering this question?
G, we have to look at the structure of glutamine and see like what are the strongest intermolecular
So
forces, right?
it is something about glutamine, right?
Right.
Which of these answer choices best talk about glutamine?
It's polar, so it's, so the thing is I saw the answer pre-populated to C, so I'm trying to make sure I understand every other option.
This is a very good thing to do, yes.
So steric hindrance would probably cause it not to be rigid, so you can eliminate it based on A, and then B, it's nothing's charged in glutamine, so that's the elimination there. D, so that's the thing, I don't quite know what they're meaning by pi stacking. But it's, yeah, can you define what that is?
Of course. So bistacking takes place when you have a lot of aromatic rings, and it takes place between mostly nonpolar functional groups. Since glutamine is a polar, it's less likely to do bistacking.
Okay.
One thing I'd like to mention is Steric hindrance due to size does cause rigidity.
Because of van der Waals?
Possibly. But it's mostly due to size, because if it takes so much space, it cannot move a lot. Like, think about,
Proline, right?
yeah, it's like it's just sticking out, like it's in the place of everything else. So you really cannot move around much.
Okay. Okay.
So which of these answer choices best represents the group of glutamine?
Yeah, I would just pick the cross elimination C. But then with glutamine also, it's like the lone pairs on the side chain, right? Can
Yeah.
hydrogen bond, and then it's there's also the oxygen around. Oh, it can the carbonyl. Sorry, the amide group, the oxygen in the amide group. Right, can it act as a hydrogen bond acceptor?
So something about hydrogen bonding, right?
Yes.
Okay. So the thing is C or A or D?
See.
Yes. Yes. All right. So the answer is C, which is right. Okay. To answer your question about hydrogen bond acceptor, let me find a good question for you. Good image for you. I'm sorry. All right. It's just gonna take me a second to find the image. All right. Hopefully this one opens. Okay. Do you see this?
Yeah.
Okay, great. So I'm gonna make this bigger just so you can see it. So basically, the difference between a hydrogen bond donor and a hydrogen bond acceptor is that the donor has the hydrogen. The donor gives the hydrogen because it's hydrogen bonding. The acceptor has the lone pair. So you're right. Does it serve as an acceptor or a donor? So let's look at the structure, okay?
Wait, can I draw it out?
You want to?
Yeah, it's okay. I think I can.
I don't think you have to memorize a lot of the structures for the MCAT though. You see what I mean?
Oh, yeah. I'm trying to. I just want to. Oh, I can't. I have to start a new. Okay, this is too complicated.
You're trying to figure out how to draw it.
Yeah.
Yeah, we
Let's
gotta
see.
start a new tab to do it. Oh, you already did. Okay,
Is it
let's go
working?
there. Yeah, let's go there. Let me share my screen so that you can. Okay, go ahead.
Okay. This is the first time I'm doing this, and I'm kind of dreading it already. I should. Okay, these should be like charged, but I'm. So if I'm interpreting this properly, this, okay, this can bond, this can form a hydrogen bond this way, right?
Correct.
Okay, so I'm interpreting, okay. Now, I guess my question was, this like, the oxygen here can still also hydrogen bond with water?
Mm-hmm.
Okay, so we have, okay. So, but these also operate as acceptors, right? So if there's like a water with the oxygen here, right? This can,
Yeah.
okay,
So basically, the hydrogen here coming off the amine group can
yeah,
serve as a hydrogen bond donor. The lone pairs here can serve as a hydrogen bond acceptor.
Okay,
So
got
it
it.
does serve as both hydrogen bond donor and acceptor, but most importantly, it does contribute to hydrogen bonding.
Okay, sounds good.
Does that make sense?
Yeah, I wanted to sanity check.
Oh yeah, of course, of course. Um, do you have any questions that we can work out through together?
Um, oh god, I should have prepared this.
Oh, no worries.
Let's
This is just anything that has on your head. Just I'm happy to help.
uh, I guess just in general with biochem, like if I had to tier, like if you had to prioritize, like what are the more high yield concepts? What would you say? I know amino acids, that's pretty much you have the like the protein structure, those are really high yield. Um, in terms of the pathways, would you say it's worth memorizing all the glycolysis steps or just memorizing the major products and the rate limiting steps? And same thing with pentose, phosphate pathway, and gluconeogenesis. What are the like? I don't know. I feel like it's a lot of work to remember all the steps when it doesn't seem to be popping up as much as like just the basic products
Yes.
and the overlap.
This is a very good question. So if you like, if you get a questions about glycolysis, if you get a
Yeah.
question about pentose phosphate pathway, 95% of the time it's going to be about the rate limiting steps, the major products, the major reactants. So like how many ATPs they invest in glycolysis, how many ATPs they get out of it, how many ATPs you get out of pentose phosphate pathway, what are the major products of it, what is the function of it in ATPH, stuff like that. The same thing for the Krebs cycle. The Krebs cycle, memorizing a mnemonic for it of these steps, it doesn't really hurt. You don't have to know the all the structures of everything there. This is just a lot. You don't have to know every enzyme name. This is a lot. So focus on the rate limiting steps. Focus on what do we get into it, what do we get out of it in terms of molecules, and what we get out of it in terms of function. Why does it happen? What does it help us?
So
The Yes.
oxidizing and reducing agents too, right?
What do you mean?
Like NAD versus NADPH and like how many like FADH2 molecules come out. I'm just worried based on some of these third-party tests. Um, and I'm asking a bunch of questions. For the electron transport chain, do we need to know the order of the electron acceptors?
I don't think that level of detail is really important. If you get a question about metabolism, like it's going to be focused a lot on experimental analysis. They're going to ask you, did you understand the passage, right? You're going to use very basic background to answer the questions. I think if you had the chance, if you had the choice between using your time memorizing the direction of steps or doing additional practice passages, I think doing practice passages would be better. So memorizing the order of when each oxidizing reducing agent is used might help, but I don't think it's the best use of your time. Does
Okay.
that make sense?
Yeah.
So it is very different than how test us on this one in undergrad, right? For now, we really want to focus on like the big picture. There's something I forgot to mention is how are these major pathways regulated, especially with regards to insulin and glucagon. So if you're fasting, if you just ate, what happens to those pathways? Does that make sense?
Well, maybe let's
Do,
talk about it right now. Fasting is glucose levels will go down, so you're less
yes,
likely to break it down. You want to
yes,
produce it. Glucagon goes up,
yes,
and my God, uh, and then gluconeogenesis is going to actually, it might actually happen because you need more
you need
glucose.
glucose. That
So
is
and
correct.
glycolysis will go down, which
Yes.
might also bring down cellular respiration with it.
You
I
have
think
cellular respiration. I think it
it's
was something.
dependent on the glycolysis products, right? So would you say that kind of is downstream?
Just keep it very general. If you don't
Okay.
have enough glucose, you want to make glucose, so you can have more gluconeogenesis.
Where does pentose phosphate pathway play in all this?
The role, what is the role of pentose phosphate pathway, do you think?
I actually don't know because I haven't studied it yet.
So yeah, just focus on those. So I have to refresh my memory on those because I took the MCAT like last year. But based on what I remember, the pentose phosphate pathway helps forms different forms of sugars. So like 4-ring structure, 3-ring structures, 5-ring structures. So it helps with producing sugars to be incorporated in forming nucleic acids. The second function for it produces NADPH.
Okay.
Yeah.
All right. Let
So yeah,
me take
but
another.
like most importantly, do you see how I'm going big picture? What does it do? It does like it helps nucleic acids and produce NADPH. What are the major input? What are the major outputs? This is what we want to focus on for the MCAT, especially about metabolism.
Right. I'm just really worried because I feel like the content review is so broad. They'll just pick a little detail from one of the books and like plop that into a question. And it's a matter of just prioritizing my brain memory for what matters.
If there is a question that asks you about a very, very small detail, like what is the structure of this enzyme? And like, oh my God, why didn't I remember the structure of this molecule, whatever. It's in the passage.
Hmm.
So I think prioritize your time on practicing. Uh, if you understand the major ideas, major input, major output, rate limiting steps, how are these regulated generally? If you're fasting, if you're hungry, with insulin, glucagon, these major ideas, you should be able to answer questions. So you should start doing practice problems.
How did you so how did you do your content review?
Um, I made a mistake. I
Oh,
spent a lot of time doing content, a lot of time, and not as much time on doing practice passages.
at least you're the third person who's told me that. And I won.
Yeah,
Yeah.
from what we're doing right now, I feel like you have very, very good background. So starting on practice passages, especially being able to understand figures, experiments, is what's really important for the MCAT.
Okay. Do you
Does that
think
make
that
sense?
Anki, yeah, do you think Anki review is like comparable to that, or like even with that, you just need to keep doing like even if it's a random passage to keep on doing it?
Anki depends on how you use it.
Yeah.
How you make your flashcards definitely helps. Memorizing terms you keep missing, memorizing stuff you keep missing, but making flashcards is also important. Um, I think it's a very good way to memorize information, especially the one you miss very often. Um, so yeah, I think it's helpful. But if you tell me Anki over practice passages, I would do practice passages.
Okay. And for that, you did Jack Weston New World, or was there
Yeah,
anything?
yeah,
Okay.
those are Jack Weston. Of course, the best is AMC. But
Mm,
if you're in that content building phase, it is difficult to use AMC because their explanations are not the best. They can be like answer A is right because answer A is right.
mm, okay.
So yeah. So but if your content is solid and you just want to practice your passage reading skills or critical thinking skills, definitely the AMC is the best resource because your exam is going to be AMC. It's not going to be U World. It's not going to be Jack Weston. It's going to be from there.
Okay. Okay. Sounds good. Can I message you if something comes up?
Of course. Do you have me on
Yeah.
Slack?
I'm on the Slack, so you should be there, right? I'll find you.
Yeah. My name is Malaz, so I should be there.
Okay. Okay. Sounds
All
good.
right. Any other questions?
Not at the moment. I will have many. Do not worry.
All right. No. feel free to message me whenever you want. So then with this, again, reach out to me via email, like the email that I mentioned in the beginning, info@premedpeers.org, or you can Slack me if you're in the Slack group. So I will end the recording really quick. All right.